And so picking up where we left off there, lipids, which are fats and oils, are not actually soluble in water, so they're what we call nonpolar or hydrophobic, water-fearing. So some other examples are phospholipids, and remember that is what makes up the cell membranes in every single cell. So fats can be used for energy storage, and there are two types of fats, and they get their names based on their molecular structure. So saturated fats are this example of fats on our left here. So this type of fat, these are actually called fatty acid tails right here, and notice how these tails are stick straight, and that's because if you look up here of how it's carbons and hydrogens are associated. So since these tails are stick straight, these are the type of fats that are solid at room temperature, because when you pile a bunch of these together, you can get them really close together and not a whole lot else is going to fit through them, which is why if these are in your arteries, that is going to impede blood flow. And versus unsaturated fats here over on the right, that if you look at this little diagram, these fatty acid tails are called kinked, and so because they're kinked, these molecules can't get as close to each other as the saturated fatty acid tails can get. So because these tails are a little bit farther apart, the molecules are a little bit farther apart, so at room temperature these fats are going to be liquid, like this example down in here of olive oil. So your saturated fats come from animals, and your unsaturated come from plants. So if you think about the types of foods that you eat, if a type of fat is liquid at room temperature, then that would be a good fat because it is unsaturated. And when I say unsaturated, I mean that tail right there has not as many hydrogens as the saturated tails have. So the saturated fatty acid tails are saturated with none other than hydrogen. So to wrap up chapter 3, I want to bring you back to one of those first documents that I was telling you about that I haven't shown you yet. And that document is called Chapter 3, Molecules of Life. And it contains several questions for you to stretch yourself a little bit in applying the ideas from chapter 3 into a little bit deeper level of understanding. So I'll pull those up for you right here. So here is that document for you. And I'm back. The first question I want you to think about is whether or not hydrolysis of a protein requires an enzyme, just like dehydration does. So think about enzymes as being catalysts, or kind of like the gun that goes off at the beginning of a track race. They get the reaction started, so think of it in terms of those hints there. The second question, of the 5 functional groupings of proteins, are any of them not present in your little finger? So look at your little finger and, do you think that there's any chemical processes happening in there that require catalysts, or what we call enzymes? And then structural proteins, well, that would be, for example, the keratin that makes up the outer layer of your skin, or that makes up your fingernails. Contractile proteins, those are types of proteins that actually work your muscles. So if I can move my little finger, it must be because I've got some muscles doing something. Now maybe there's not a whole lot in my little finger doing heavy lifting, but I'd be willing to bet that there are some in there. Transport proteins, those are ones that we can find in the cell membrane, and they actually transport different sizes of molecules into and out of the cell. So if you think those are in the cells of your little finger, then we might be onto something here. And defensive, are there any defense proteins? So that would be proteins involved in immunity and dealing with your white blood cells, Your next question here, why don't the amino acid groups of amino acids make all proteins polar? And that is something that you can actually look up in section 3.2. Next question, why do you think most heat denatured proteins don't spontaneously refold? That's a great question from that same section 3.2. Using the letters C, A, T, and S, I believe that I meant to put G in there. Write as many 3-letter words as you can, and what I want you to do with this exercise is to define each word, even if it's a silly word that you make up, or if it's an acronym for something. And what I want you to do with this exercise is to see how making a bunch of 3-letter words, I don't know if you can see the ones I did here. Where are they? I made a bunch of different 3-letter words. Right there. And so you're going to use just those 4 letters, just like there's 4 nitrogenous bases in DNA. And with these 4 letters, C, A, T, and G, you can make up a whole bunch of different words and define them differently, just as how in DNA, A, T, C, and G make up every single trait that you have physically, and some of your non-visible traits as well. So the next question, if one chain of DNA has a certain sequence, what would the complementary sequence be? So with that question, you're just going to work on base pairing. So for the first letter A, remember that A or adenine always complementary base pairs with T or thymine. And section 3.3 in your book is helpful there. The next question, why can you digest starch but not cellulose? Think about chemically what we would need in our bodies to be able to digest starch versus cellulose. I mean, if you ate grass, what would happen to it? Would it get digested? Would it just go through your body? And why is that? So section 3.4 is a great place to look there. And 3.5, if you hydrogenate, which means add hydrogens to an unsaturated fat, does this treatment make the fat more solid or more pliable or flexible or with room or space in between those fatty acid tails? And you can go back to, you can go back to section 3.5 and then also earlier in this video to find examples that will help you with your answer for that one. I hope this was helpful to get across to you chapter 3, and I look forward to looking at your work that you're going to produce this week. As always, please let me know if you have any questions about the videos or any of your assignments. So have a great rest of your week. Thanks.